Okay, Chapter 8 in the Green Book is a combination of mitosis and meiosis. And full disclaimer here, it drives me a little crazy to combine the two. If you notice, the Red Book and Basic Biology has them in two different chapters. In fact, the Red Book is a separate unit. And even though it's very convenient to combine the two because they both talk about cell division and they both start with the letter M and have an "its" in it, they're very, very different things. So these are going to be divided into two videos. This video is going to be mitosis. Mitosis is what your body is doing right now. You're sitting at your computer watching this, or your little iPhone, or whatever it is, and you are shedding skin cells everywhere, and to replace those skin cells, your body does mitosis. Meiosis makes babies, and you've taken enough biology to know that those are two wildly different processes. So you're going to find the videos for this divided into mitosis, this first video, and we're going to talk about what goes wrong with mitosis. You end up with cancer. And the second video, we're going to talk about meiosis, and when that goes wrong, you're going to end up with genetic mutations. Okay, so for both mitosis and meiosis, cells only arise from pre-existing cells. And so cell division is at the heart of reproduction of the cells and to reproduce the organism. So again, here we've combined the terms mitosis and meiosis. Reproduction of the cells is mitosis, and when an organism like us reproduces, we're making sex cells, we're making gametes, we're making eggs and sperm. It's totally different. That's a separate video. When we talk about the cells only coming from pre-existing cells, if you're trying to reproduce cells in the lab, you'll hear a lot of talk about stem cells. And the reason is you can take a stem cell and you can reproduce it into other cells. Where something that's already a nerve cell, it already has a job, it's not going to change its job anytime soon. But a baby cell, like a stem cell, doesn't have a job yet. You convince it to be a nerve cell, you can convince it to be a liver cell, or whatever you need in the lab. So that's where stem cells come in. It's taking pre-existing cells and making more cells from them. Animation. Alright, so here's your prokaryotic cell, and it's going to go through binary fission, and the first thing it has to do is replicate all that DNA. And sounds simple, but replicating DNA is a fairly complex process. So it replicates its DNA, and then it needs to make sure that that DNA ends up equally in both of the new daughter cells. So how does it do that? Very simply, it's going to attach the DNA on either side of the membrane. So it's going to attach here and here, and then as it elongates and pulls apart here, it's going to get an equal amount of DNA in each of the daughter cells. And that's kind of the key here, is to make two equal cells. You wouldn't want a cell with all the DNA, and then you would just have, you know, a blob of a cell that's non-functioning. So that's the trick. And while this is simpler than mitosis, it is still not necessarily a simple process. All right, so in eukaryotic cells, now we're going to get way more complicated. When things go wrong in mitosis, they can go very, very wrong. So cells are very, very careful with their DNA. We are going to duplicate the DNA and carefully divide the genes. There's more DNA than there are in prokaryotic cells, and bacteria tend to rapidly divide, and really who cares if one dies somewhere. But when something goes wrong in a multicellular organism, you're going to end up with cancer. So mitosis is a complicated but very structured process. Okay, so individual chromosomes contain a very long amount of DNA, and it's really, really long. It's several meters tall for each cell, and then you got, I don't know, trillions of cells. So there's a lot of DNA in you that needs to be replicated as your body goes through this, each individual cell. And the DNA becomes visible when the cell is in the process of dividing, and the reason it becomes visible is that when it's not dividing, that DNA is all unwound and it's being used and being made into messenger RNA to make proteins. But when it's dividing, it wraps it all up into packages to be careful with it to make sure it gets divided equally. So it's no longer going to be in use during mitosis. Okay, so before we do the mitosis part where we're going to divide the cell, the first thing that happens is you have to replicate the DNA. And so for eukaryotic cells, you're going to produce sister chromatids, and they are going to be attached at the centromere. And they basically look like little X's, but it guarantees 2 sets of those genes. So we're gonna make sure that you have produced these sister chromatids prior to the cell dividing. Okay, and then cell division involves the separation of the sister chromatids and results in 2 daughter cells, each containing a complete and identical set of chromosomes. So the first thing you have is the before it even starts dividing, before the cell starts dividing, we duplicate the chromosome and we get chromatids, sister chromatids. That's just this thing twice. And then we pull it apart at the centromere and we make sure we have 2 copies of it. And yeah, that's it. Okay, mitosis is when the cell multiplies. So you look at the mitotic phase and you go, wow, that's really a small phase in the cell. Look at all this interphase. Oh, that's a lot of cell life. What's the cell doing there? Well, the cell is doing its job. Whatever job that cell has, whether it's a skin cell acting like a skin cell, liver, blood, whatever that cell is, it's doing its job. It stops doing its job when it goes into mitosis. So you wouldn't want all your cells dividing because the DNA shuts down and it stops doing its job. So the interphase is what it's called, is the cell's doing its job. The DNA synthesis occurs during interphase, and then you have what's called a gap 1 or growth 1. It's technically gap 1, but you can think of it as growth phase 1 and 2. And then it goes into the mitotic phase. The mitotic phase can be very, very short. It can do this in 20 minutes because it can't spend all day dividing. It needs to do its job. All right, during interphase, chromosomes duplicate and cell parts are made. So what are the cell parts? We're talking all the organelles. The chromosomes duplicating was in that S phase. And then during the mitotic phase, the duplicated chromosomes are evenly distributed into the 2 daughter nuclei. So we're going to take our 2 chromosomes, like you saw in the picture. You're going to divide them into equal chromatids, the little X, and then we're going to pull them apart right at the centromere, making sure we get 2 equal chromosomes going into the daughter cells so that they are identical. So cell division is a continuum of dynamic changes. And yes, of course it is, because it's going to occur fairly rapidly, as in you could watch this under a microscope. 20 minutes, hour, and it's done. So we're going to just, it's going to change, it's going to divide, and then it's going to go back to doing its job. So the first thing that happens in mitosis is that, well, the chromosomes have coiled up, and then you get a mitotic spindle that moves them to the middle of the cell. All right, then the sister chromatids separate and move to the opposite poles of the cell, and we're going to give you all the phases for this. But basically, we're just pulling them apart here. And then you have something called cytokinesis, where the membrane of the cell actually pinches in and the 2 cells pull apart. And you're done. All right, so here we go, the stages of cell division. And interphase is that long, long phase I was talking about where a cell is a cell. It does what it's supposed to do. It has gone through the G1 phase, and then in the S phase, it has replicated its DNA. It has gone through the G2 phase, and near the end of the G2 phase, you get these centrosomes that are going to now appear. These are there at the end of the G2 phase. They are going to produce the mitotic spindle, those microtubules that are going to pull apart the sister chromatids. So, and the DNA here is still unwound. So before we destroy the nuclear envelope and expose that DNA to the nasty cytoplasm and all the possible things that could go wrong, it gets wrapped up tightly around histones. It gets completely wound up, tight, packaged up, and put into its chromatid pairs so that you can pull each one apart. That's in prophase. You can see the early mitotic spindle developing. This is what's going to pull those chromatid pairs apart, those sister chromatids, is that spindle. And now we're going to dissolve the nuclear envelope. And so that's in pro-metaphase. And you can see that the spindle microtubules here are getting ready to pull apart the sister chromatids. And some of them are going to latch on to these kinetochores, and some of them are going to help actually pull the cell apart. So these little microtubules are a really pretty important part of this. Alright, so metaphase, the way I remember metaphase is middle. That's what I think. It just helps me remember it. And I remember it that way because you can see that the sister chromatids are all lined up in the middle, like down the equator of the cell. Then you have anaphase, and that's when each one grabs this and pulls it to the other side. And this is what makes sure that that DNA is exactly equally divided. Hugely important for mitosis. And the last phase is telophase and cytokinesis. And all that we're doing here is we are going to reform these cells. So we're going to finish the elongating of the cells. We're going to have a cleavage furrow, and we're going to complete the two daughter cells. Alright, so the cytokinesis, this is an animal cell that you see here, and what you get is a cleavage furrow, and that's the first outward sign of the cell membrane that we're going to now pinch off two different cells. So that cell membrane is going to come in. Actin and myosin are the proteins that are the movement proteins that are going to cause this pinching action to create two identical daughter cells. Okay, so in plant cells, mitosis ends a little differently. We've still done all the usual stuff. We've replicated the DNA. We've pulled it to the two ends. But now we can't have a cleavage furrow come in and pinch off two separate cells because there's a whole cell wall that's pretty much fixed in place. So they do it a little differently. You can see here, you can see the parent cell wall is here and down here as well. And then what you see here are little vesicles that are going to rebuild, or to build, I should say, a new cell wall, and that is going to expand out into what's called a cell plate. And the cell plate will ultimately be the new cell wall, and you will have a daughter cell on this side and a daughter cell here. I think you can see the daughter nucleus on this side as well as on this side. So you're going to get a new cell wall right down the middle here. Okay, so when we talk about cell growth, we have to talk about things that stop cell growth as well. Cells don't grow constantly. If they were constantly doing mitosis, they wouldn't do what they were supposed to do. And if they never died off, they'd be something called cancer cells. And those are obviously really, really bad. And cell growth is highly regulated. It's regulated at several spots, and it has different environmental factors that regulate it. The thing called anchorage just means that the cell has to be anchored, literally sitting up against something. This could even be, a petri dish, but as long as it's anchored, it's going to divide. Cell density is how many cells. Do you have way too many cells? Are they starting to divide on top of each other? In that case, it's cancerous, because normally cells will stop dividing when they all touch each other. Then there's chemical growth factors, things that will affect cell division. That can be released in your bloodstream by platelets that cause cell division. If you're injured and you want to know how do those fibroblast cells know how to start pumping out collagen and fixing your body, Those are chemical growth factors. Okay, so most cells are not dividing. They only divide when they are stimulated to do so. Okay, so here we go. Most normal cells divide when they are attached to a surface. That's what we were calling anchorage. All right, so you see here that the cells are attached to a surface. So good, they're happy to divide. Then they all are touching each other here, and you have a complete single layer, and then this is called density-dependent inhibition. What that means is there's enough cells, they stop dividing. They sense through chemical sensors what other cells are sending out, and they know to not divide. If you pull a few out and there's room here, they will fill that in, and those are normal cells doing normal cell division. Okay, growth factors are proteins that are secreted by cells that stimulate other cells to divide. So normally you can see these cells have formed a single layer and they've stopped dividing. That means the cells around them are no longer secreting this growth factor. It encourages the neighboring cells to divide, and they stop doing that. They are now the density-dependent inhibition, right? They've stopped growing. We can artificially add in growth factors that will stimulate them, even though they shouldn't be stimulated. We can artificially do that, and they will continue to go through mitosis and divide and essentially overpopulate the area. So growth factors signal the cell cycle control system, and growth factors are proteins, and proteins will control the cell cycle. They will put a stop on the cell cycle, often in the G1 phase, and then sometimes right before or after mitosis. But the big one's usually in the G1 phase. And these are often kinases. And if you remember that the kinase, the term kinase means that it's going to add a phosphate group to a protein. So that is how it controls the cell cycle. All right, so here you go. Here's the checkpoints of the cell cycle, and if you get through the G1, you can go into something called the G0 phase. And this G0 phase is actually where most of your cells are in your body. Most cells are not dividing. They're not in this G1 as G2 phase. They are simply doing what they are meant to do as cells. Skin cells, liver cells, whatever they're doing. They're doing what they're supposed to do. Nerve cells spend their entire life in this phase. They tend not to replicate. And so you can get kicked out into this G0 phase. You can also get brought back into the mitosis phase if you add a growth hormone. We can do this artificially in the lab. The body can do this as well. So you have the first checkpoint, this G1 checkpoint. We stop mitosis, and we allow the cell to stay in interphase, again, doing what it's supposed to do. If and this is where it mostly stops, but if this is passed, okay, so that protein is not present, that kinase is not there, and you replicate the DNA and you go to G2, there's another checkpoint that can occur before cell division, and then another one afterwards that will stop the daughter cells from going through this phase. So there's multiple checkpoints that have to be passed, and all this is to ensure that your cells don't become cancerous and you don't get tumors overgrowing your body. All right, the two types of regulatory proteins are these kinases. Remember, they're going to pop a phosphate group on, and something called cyclins. And cyclins are another protein, and they're literally called cyclins because their amounts in the cell, how much cyclins you find in the cell, cycle with the cell cycle. So they got the name cyclins. Quite inventive, isn't it? So when the cell is going to move into mitosis, you will find more cyclins. They then activate these kinases. So because the kinases depend on them, and these, remember, they just add a phosphate group, phosphorylate something, they're called cyclin-dependent kinases. They are controlled by the cyclins, which will fluctuate with the cycle of the cell. Okay, So the number of cyclins rise and falls during the cell cycle. Cyclin activation requires phosphorylation by a cyclin-activated kinase and binding of, ready, number 1, at the G1, the proteins produced in G1 bind to the cyclin E and the cyclin-dependent kinase 2 proteins and activate it. That causes the cell to enter the S phase. Yes, I know you're writing all of this down. At the end of the G2, the binding of the B cyclin, okay, cyclin-dependent kinase 1, so CDK is just cyclin-dependent kinase, and the mitotic cyclins trigger entry into M, which is mitosis. This phosphorylates proteins needed to enter the M phase. So it all depends on the cyclin. The kinases, these cyclin-dependent kinases, they just hang out pretty much all the time in the cell in equal numbers. They don't go up, they don't go down. It's the cyclins that are going to come in and activate these that'll cause the transduction pathway to occur essentially and cause this to go into the M phase. All right, so let's look at one of these checkpoints. We're going to look at the checkpoint that is at the end of the G2 phase. And it is a cyclin-dependent kinase. It's a specific one called the maturation promotion factor. This is in your book, so if you're trying to take notes on this whole slide, you can look this up. What I want you to note is that as the cyclin fluctuates here, so does the MPF, this maturation promotion factor. And what it does is it allows the cell to go from the G2 phase into mitosis. Without the cyclins increasing, the mitosis won't happen. The M phase will never happen. And you can see it down here. Normally, in the G1 phase, the cyclins are degraded. They're not in the cell. That's why they're called cyclins. They cycle. So G1, they're degraded. They're not there. After the S phase, they start producing the cyclins. You have this maturation promoting factor, and the cell can go through mitosis. All right, so binding of growth factors to specific receptors on the plasma membrane. This is very, very familiar to you. You have the same kind of transduction pathway that you usually see. You have a growth factor. This can be from platelets in the case of an injury. There are specific growth factors called PDGF from platelets. But any growth factor that comes to the cell, binds to the receptor, you do the same old signal transduction pathway, and that will allow the cell to go through mitosis. This is the G1 checkpoint. This is the big checkpoint I was talking about earlier, the one that most cells wait for. They wait for some kind of growth factor. They don't normally just do mitosis randomly. They're waiting to be told to do it. Okay, so if a cell starts growing out of control, it can become a cancer cell and produce malignant tumors. More often than not, cells that start to grow out of control are destroyed by your immune system. A healthy immune system is designed to seek out and destroy precancerous cells. But when that doesn't happen and the cells are going to grow out of control, they become cancer cells. And they're going to divide excessively. They're going to divide and divide and divide. There is some cancer cells in a petri dish, and I can't remember where, but they've been dividing since the 50s nonstop, and they don't die. Normal cells will die. Cancer cells will just keep going through mitosis over and over and over again. Okay. A malignant tumor can invade other tissues. The thing with cancer cells is they're not necessarily dependent on being anchored. Remember we said other cells won't divide if they're not anchored? Cancer cells don't really care. They're just going to keep dividing, and so they can be thrown out through your lymph nodes and into blood vesicles, and it doesn't matter to them. They're going to divide and divide and divide, and that is how cancer can spread through your lymph node system to other parts of the body. So here you have obviously a breast cancer or a tumor, and as it grows, it goes into the lymph nodes. If you know anyone who has breast cancer, they will often, or always I should say, test the lymph nodes to see if there's cancer cells present there. If there is, there's a good chance it has metastasized into the rest of the body. Radiation and chemotherapy work because what they do is they find and kill the cells that are dividing the most rapidly. So especially chemotherapy will hit skin cells, so it'll hit its target cell, the cancer, but people going through chemotherapy will lose their hair. The reason is follicle cells tend to divide very rapidly. They'll often have skin issues, rashes and that kind of thing, and that's because skin cells divide rapidly. They very often will have digestive issues, and that is because, again, the cells in your digestive system divide rapidly and therefore are also hit by the chemotherapy along with the cancerous cells.